Just read it out of the textbook. Yeah, so it's like I didn't comprehend anything.
All right. So let's see what kind of stuff you came up with. Stuff you've heard of, stuff you think of. This is brainstorming. There's no wrong answer. So we're just going to go around and see if you can just share like one thing that you guys talked about. We'll start with you guys.
Yeah, we said like you could either make it like perfect or like changing diseases and like traits you have in your genes.
Okay, so changing traits, changing diseases. What did you guys talk about?
We said the first thing that came to mind was like GMOs, like
Yep.
We talked about Elon Musk is trying to put like the chip in your brain, and you can make it like send signals to certain like nerves and parts of your body, so you can like fix paralysis apparently.
I know that. Yeah, I don't know a lot about that. I know they're trying some of that.
And like cure diseases that haven't been cured before, just because like you can tell your brain something else, so like just fix it.
So things mostly related to the nervous system and like your brain and stuff, right? Probably because it's all like electric signals.
We talked about like ethical considerations, genetic engineering, and whatnot.
Okay. Yep. And there's a lot of ethics stuff that comes into play anytime you start talking about messing with DNA, right?
We talked about how you can probably work on changing genetics.
We talked a lot about what books we talked about, like the perfect baby and how you can change the DNA of the baby before it's born.
Yep.
We kind of mentioned like criminology stuff. That was one thing that you said.
Awesome. Yep. Forensics and crime scenes and stuff. People online said a few of those same things. Kylie also mentioned things like mapping out DNA with all of the nucleotides and genes. And Ashley mentioned CRISPR. If you've heard of that, it's a method for genetic engineering that we'll touch on a little bit later. And the designer baby idea, with which a couple of you had mentioned. So they talked about kind of some of the same things there that you had mentioned. Obviously, biotechnology is a really new and growing field of research, right? We're continually developing new techniques for changing and editing genes and coming up with new ways that it can be used. So we're just going to kind of scratch the surface here. We're actually going to come back to some other biotech related labs later in the, I don't even remember if it's this trimester, third trimester, I think, and talk about some other techniques. But there's tons of stuff that we're not going to get to because this is such a huge and expanding field. But we're just going to talk about a few of the techniques that are out there that are pretty commonly used in a lot of different ways and some of the possible applications. So as we start talking about this, there are a few terms that you'll hear. There are definitions of them on this slide. They are all slightly different. You don't need to know the differences between them really because they overlap a lot, and we'll use these words pretty interchangeably when we talk about them in class. DNA technology specifically looks at manipulating and sequencing DNA. Sequencing just means literally figuring out the order of the A's, T's, C's, and G's, the bases that make up the genetic code, and kind of reading the code. Biotechnology is focused more on manipulating organisms or things to make useful products. So if you're thinking about genetically modified crops, like foods that are more disease resistant or things like that, that's biotechnology. Genetic engineering is more directly manipulating genes for a specific reason. That would be something like fixing a mutated gene to cure a disease. That would be an idea of like genetic engineering. where you're changing the DNA for a specific reason. And obviously, like I said, these overlap a lot. You don't need to worry so much about the distinctions between them, but I wanted you to kind of see that there are slight differences in these. And as you already mentioned, there are a lot of applications for these. You talked about GMOs, you talked about forensics and criminology a little bit, things like paternity testing. We can use this to manufacture products that can be used to help treat certain conditions. Like we can manipulate genes that code for certain proteins like insulin that we might need. And then we can collect the insulin that's made by those cells and use it for people who need additional insulin, for people who have diabetes. We can also use this just to identify genes that cause diseases, to figure out if people have a gene that could cause disease. Another application for some of these that's kind of related to that last one is right now with the COVID pandemic, a lot of scientists are working on sequencing the genetic material of different strains of the coronavirus that we've all been hearing about and living for a year. You've heard about all the new variants coming out, things like that. Scientists, if they go in and actually sequence the genes in all of these different types of viruses, they can find where the mutations are, and sometimes they can kind of trace it back and figure out where that new variant started and where the mutation happened. And that can kind of help them map the spread of kind of where this virus is going and how it's changing. So there's a lot of genetic technology that's being used to help us track and learn about the pandemic right now as well. We'll talk about some of these a little more specifically with some of the lab scenarios we do over the next few days. Other ones we won't really spend quite as much time talking about. And obviously this is not an exhaustive list. There are more applications than these. And the more we develop these types of genetic technology, the more things that we find to use them for as well. So I added this slide in here just to kind of give you a summary and an overview of the main ones we're going to be talking about this week. So you can write this down if you want. Most of these will also be discussed later in your notes, but this is just kind of the overview. The ones we're going to be focusing on this week is we'll talk about the process of DNA sequencing, how we actually go through and kind of read the sequence of A's, T's, C's, and G's in order to figure out the genetic code of a particular organism or even just a particular like disease-causing gene, but find that actual sequence. We'll talk about these things called restriction enzymes, which we use to cut up DNA into pieces. We'll talk more about why we would want to do that as well. This is one of the ways that we can help identify where a certain sample of DNA came from, because restriction enzymes cut DNA at a certain base sequence, and since everyone has slightly different DNA, those sequences occur in different places. We'll also talk about a process called PCR that stands for polymerase chain reaction. You don't need to write that down right now because I know that's in your notes later. This is a way scientists can make many, many copies of a gene. And that can be really important if you only have a small sample of, say, a gene from a newly discovered species or a gene that was found at a crime scene, a small sample of DNA, but you've only got a little bit of it. It's good to have lots and lots of copies of that genetic sequence so that you can work with it in different ways. And the main one we're going to focus on this week that we actually have the equipment and stuff for in class is gel electrophoresis, which is a way that we can separate DNA. This can also be used with other molecules like proteins, but it separates molecules according to their electric charge and according to their size. So most of the labs we're doing this week are going to use gel electrophoresis, and we will actually be using some restriction enzymes day after tomorrow, I think, maybe, as part of one of these labs as well. And I'm going to show you just a quick animation to kind of review what electrophoresis is. Do you guys remember doing electrophoresis in 10th grade? I think most people did it. but sometimes depending how much time we have, it's pretty brief. So I just want to show you this animation real quick that goes through and has some funky sound effects, just so you know, like that. So it starts just by kind of setting the scenario out, saying you're holding this little plastic test tube. It's got some DNA of different lengths, and you want to measure how long those strands of DNA are, because sometimes that can be a helpful identifying feature. After you've cut up some DNA, you measure the lengths of the strands, and that can help you identify someone. And we'll talk about exactly how that works a little later. And so obviously, if they were actually visible, we'd be able to just organize them, and that would be really easy. But they're microscopic and we can't see them. So gel electrophoresis gives us a way that we can separate these strands without actually being able to see them. And so this is kind of how this works. It's called gel electrophoresis because we use this agarose gel that you can see is made up kind of like a sponge of Jell-O. And this is actually a little bit of one that I made when I was preparing for this. And if you want to touch it, you can. This one ripped, so it's only like a quarter of it. But you can see it's kind of like Jell-O. It's very similar to the stuff that you grew bacteria on in the past, probably. And you can also see that as I made this gel, I put a bunch of little holes in it called wells. Those holes do not go all the way through. They're just like little divots in the gel. Okay, so the Jell-O itself is kind of like a sponge. It's got all these little passages and holes through it. The little wells are where we put the samples. So as we go through here today, you will be practicing putting the samples into the wells, because as you might have noticed, those wells are pretty tiny, and that's a little bit tricky. And so we're going to practice that part today. And then what we do is we use an electric charge to cause the DNA samples to move. Take a minute and turn and talk to your partner. What's the charge on a DNA molecule? And how do you know it has that charge? Why does it have that charge? I'm going to go back once just so we don't have to listen to that sound. Hang on.
Yeah.
So here's a hint and another review question. Talk with your partner. What are the 3 parts that make up a nucleotide of DNA? Okay. So I think I heard most people. I heard base and sugar. And what's the 3rd part of a nucleotide? The phosphate. Base, sugar, and phosphate. Do we remember that? Kind of, sort of. What's the charge on a phosphate group? Negative. Phosphate groups have a negative charge. And since the entire sides of a DNA ladder are sugar, phosphate, like there's a lot of phosphate in DNA overall has a negative charge. So when we run this electric current through it, if we put the DNA samples here, the DNA is going to be repelled by the negative charge and attracted towards the positive end. And that's what causes it to move through the gel. But the pieces don't all move at the same speed because they have to kind of wiggle their way through that gel. The way that I think about this, have you ever been somewhere really, really crowded with a very small person? Do you know how quickly a very small person can move through a crowd? Like they can just zoop and they're gone. and you can hardly make your way 2 steps, right? That's the analogy I think of when I think of how this works. The shorter DNA strands can weave their way through that gelatin sponge of the agarose really quick. And so the shortest pieces of DNA go the farthest. The longest pieces don't go as far because they can't get through that substance as quickly. So all of the DNA that was here, it's all moving kind of down this row, but the little pieces go a lot faster and the long pieces go a lot slower. And so after it's run for a while, it kind of sorts itself out according to size. And so when you look at it at the end, you end up getting what they call a banding pattern. You can see these little dark bars. That's where the DNA is. And that's not a single DNA molecule. This bar is a whole clump of strands of DNA that are all roughly the same length. And these would be the really short ones. And so then if you run a sample where you know the lengths, like maybe if we knew this sample had bases that were like 100 base pairs long and 200 base pairs long, you can use that to kind of estimate, okay, this one must be around 150 base pairs long. Does that kind of make sense? So that's kind of how we run this and what we're looking at here. The equipment that we have, just flip this over here real quick. So the equipment that we have here, I'm just moving this so that they can see what I'm looking at, that we will be using this week is we have these green boxes. This is the power source where you can plug in the positive and negative end, and you plug this into the wall. I didn't bring that part up here because we don't need that part. The gel itself goes in a little tray. It's really hard for me to show you and them at the same time. There we go. So the gel itself goes in this tray right here, which then gets set into the electrophoresis chamber. And then you put the cover on it that has the positive and negative electrodes. So that runs a current. And we fill this whole thing up with an electrophoresis buffer, with a liquid that will carry electricity. Because otherwise we'd have all this space, we'd have air, and the electricity isn't going to travel real well through air. But by filling it with liquid, we kind of create a continuous path that electric current can flow through so that our DNA can travel through the gel. Does that kind of make sense? Like I said, we're going to be doing a couple different labs with this throughout this week. All right, I'm going to bring this back up so you can see me. Look how lovely that is. All right. And I just talked about that. So today, what we're going to be working on for the rest of class today is a couple of things. First of all, those of you who are here are going to practice loading some of the wells. Like I said, this takes a little bit of like steadiness of hand and a little bit of practice so that you know what you're doing. So today's a practice day so that you can actually do this with real samples over the next couple days. When it's not your turn to practice, because we're going to have to go in like 3 shifts, you'll be working on the pre-lab and reading some background information about this lab, okay? So in Google Classroom today, you will see in the homework section, there is a post called Gel Electrophoresis Part 1. There is a lab document, and there's also an additional background reading that just gives you more information in how these procedures work. You need to do that. You need to use that reading to answer some of the background questions as well. But you can start working on, I think, questions 1 through 13 today. We're going to continue working on this tomorrow, but it'll give you an idea of kind of how this process works and some of the background that you need to really understand what you're looking at in the lab tomorrow. Questions about that? Okay, so those of you who are in class, I'm going to talk to the people online for a minute. You can pull out devices if you've got them and start pulling up that lab document. And then in a minute, I'll show you where the practice things are. People who are online, that is really all that you need to stick around for today, because they're not actually setting up a lab. They're just going to practice putting the samples into the well. Tomorrow when we do a real lab, we'll show you more of what's going on. So you can go and get started on the pre-lab questions and the background questions on that lab document so you have some idea what we're doing tomorrow. And then remember, tomorrow is Wednesday, so class is at 2:25 for 5th hour. Okay? All right. Thank you for joining us. We will see you tomorrow.
Thank you. Bye.
Bye.